Hello everyone, thank you very much. Making CBD a drug. That's what I was going to talk about in 2020, and I'm really sorry to do the bait and switch on you. We were really looking forward to do that project, but a few things came up, and this is what we're going to be talking about today. So, 2022. Thank goodness. So first about who we are. We're Delic Labs. We're a contract research organisation. We prize collaboration with everyone we can. We want to try and bring as much of the scientific method as possible to cannabis. We want to try and make information more widely available. We do a lot of collaboration with the University of British Columbia. We're actually based on campus-ish. So we've got access to a lot of instrumentation that a regular lab just simply cannot afford through our industry collaborations. But we also like to do a lot of collaborations with other people in the cannabis space. Monica and Linda are two examples of that. So our research topics are chemistry. We're a bunch of chemistry nerds who that's what makes us happy. That's what we do. I'm the comp guy. My background is computational medicinal chemistry, so drug design and molecular modelling. And I'm sort of on board as data and that side of things. The rest of them are very interested in organic synthetic chemistry, analytical chemistry, and those sorts of things. What we work on is anything that is a biomolecule and is regulated. That's what gets us off. So we're lucky enough in Canada that we have what's called a section 54. Oh God, my dyslexia is kicking in. I think it's section 54, which is an exemption to do psychedelic research. So we can work with a whole range of interesting biomolecules. We're doing some DMT stuff, some psilocybin, some psilocin, and those sorts of things. But today I'm going to talk about a client project we did. Our client, unfortunately, I can't talk as much about this as I would like to. I can say our client was a multinational. We were trying to develop an enhanced CBD vape, and what they engaged us to do was a virtual ligand screening. So we're screening potential molecules in computer simulations to try and reduce their their in vitro and their in vivo load. So again, I really wish I could talk more about this, but I really can't. All I can say is that we were looking for terpenes that are expressed in cannabis, and we're looking for agonism at the GabaA receptor, and we're looking for CNS GABA. So the medics can probably have a little bit of a guess what we might be trying to do there. So first I want to talk about virtual screening. So this is the process that you would go through to determine how you're going to do your virtual screens. So the first question is, what do you know about the protein of interest? Do you have any structural information? Do you just have a sequence? In our case, we actually had some protein structural information. There are crystal structures of the GABA receptor. So that means we can go along and do some docking, which is just in silico placing ligands on the protein and seeing where they interact. And then from there, that may even answer some of your questions. And there are other routes you can go after that, which I'll take you through as we go. So, what was the plan? Well, the first step was to have a look at the existing crystal structures, see where the current molecules were interacting, seeing how we could see if we could find out how a terpene might bind in those same pockets, compare any of the ligands and to those native crystal structures, and then let's just see if we can jam some ligands in there and see if they're happy. Oh, I should point out, ligands in this case really just means small molecule. It's a throwback to inorganic chemistry where you're chelating to a metal The inorganic chemists hate us molecular modelers using this, but at least we don't call them ligands. So a little bit about GabaA, the receptor, the ion channel. The endogenous ligand is GABA-aminobutyric acid. It's a really common neurosignaler. It's found throughout the body. There are multiple subtypes, each of the subunits have different types, and then the subtypes are comprised of different versions. I'll give you a little idea. It's quite a complex protein structure. So there are these 5 protein subunits that are all come together to make a transmembrane pore. It's an ion channel. So what it does is it depolarizes synapses, which is an inhibitory mechanism. It calms you down a little bit. The important thing is this picture down the bottom. What you see here is that there are 2 GABA sites, and there is another one there that is labeled the benzodiazepine site. So that was the site that we were targeting. That's where we wanted to interact. And it's been shown that it sort of acts as an allosteric modulator. So it doesn't make the channel turn on by stimulating that site at all, but it does potentiate GABA's effect on that receptor. And the other thing to note is that all of these sites are at the interface of the 2 proteins. So you really need to be studying the complex as a whole. You can't just look at one of the subunits. Right, so now I went on and did some literature research before I jumped into the project. And what we found is that there is limited evidence of terpenes interacting with GABA and having an agonist effect, which is pretty cool. So there's some good news at the beginning. And there's a whole lot of pharmaceuticals that are well known to interact with GABA, so great, awesome. There's some crystal structures out there. What I found very quickly is that the quality of those crystal structures varies hugely. Because GABA is such a complicated protein and it's membrane bound, the only way you can really get it to crystallize well is by using some pretty unrepresentative physiological conditions. You end up throwing a lot of detergent in there to get it to actually crystallize. And what that means is that the conformations get distorted and you end up with a structure that's not particularly representative. Classically, it's down in the transmembrane domain that they actually collapse on the core and you get some rotations and some twists. That turned out to be a little bit of a problem. I'll get to that later, but the gist is that it's thought that CBD does activate GABA, and we were interested in looking at CBD at GABA as well. But it's thought that CBD doesn't actually bind at the benzodiazepine site. It binds down in the transmembrane domain on a cleft on those proteins down there. And since that area is heavily distorted in the crystal structures, it makes it much harder to model. So up here, we're looking at the benzodiazepine site here, sorry. So we've got the 2 different crystal structures here. We have 6HUO and 6DT5, I believe. In green, you can see the 6D65 structure. And where I've got that red arrow, I'm showing that the 2 structures are quite distorted in comparison. The white ribbon basically means it's forming a beta sheet. That's the sort of structure you'd be expecting. The green wiggly lines represent loop structures, which are normal in a protein. You do expect to see loops. You just don't expect to see them there. So what that's showing is that in the process of crystallizing, that structure has been distorted. Right, so on to the in vitro data. So, like I said, people use benzodiazepines. They know they work. That's awesome. Great. Woo, yay. However, most of those were developed a long time ago. And what they did is they just stuck them in humans until they saw it working. And they went, great, it's the dose. There's not easy to find out how strongly a compound binds to GABA alone. You can't really just isolate GABA, stick a molecule on it, find out what concentration it's actually binding at. Most of these assays are done through patch clamps, which is where you express the receptor in, I believe it's a frog ovary, and then you run a voltage across it. And then you're looking for the calcium transmission. So what you're actually doing is a functional assay. You're not really doing a binding assay. You're not really seeing, because you can't do a classic washout assay here. So I trawled the literature and I found a whole range of different values. I didn't find any binding data at all. I found some IC50 data, which was confusing because it's not inhibitory, so I don't know why it was labelled IC50. I found some EC50, which is what I was expecting. However, the numbers I'm showing here are averages, and they differed by orders of magnitude. So that was a problem. But, you know, we're brave. We're going to push on anyway because they're paying us, so we're going to do it. So I thought, right, well, the first thing to do is we know linalool works, so let's dock linalool. And sure enough, that's actually the GABA site. And linalool, in green, lies beautifully on top of GABA. It just sits there, and it was like, great, okay. So it binds to GABA, and then it bound to the benzodiazepine site, and it bound to about 15 different other sites. So it was like, oh, docking, right. So I can select anywhere on the protein, and I can dock linalool to it. And I was like, oh dear, so this isn't going to be selective. So how do we actually indicate what's working and what's not? So we thought, right, what we really need is some binding energy calculations. So this is a way of calculating how strongly it's docking to a particular spot. Like, does it actually want to be there? And the basic gist is that there is a state function between freely floating ligands and the protein, and then they go through a transition state, and then they collapse down into their bound conformations. They both adapt their conformations a little bit. Their lower energies change, and if you have a favourable binding, you'll have a total drop in energy. So this is molecular mechanics and generalised Born surface area approximations. That's not particularly important. All I'm here is to say is that there is a way of calculating this. You can do it with a computational simulation. It does neglect entropy, which is an important part of that equation I showed you earlier. The justification is that usually you use a cogeneric set of ligands, so a whole lot of ligands that look like each other. They've just got small variations in functional groups. And you can assume that they all have similar entropies, so you can cancel that term out across a series. I was testing a broad range of terpenes which are not cogeneric, so that assumption was a little questionable. So we did the work anyway, because you've got to see how it's working. And these are just the control compounds. So these are just the pharmaceutical compounds that we have some estimates of their binding energy predictions. And what you can see in the molecular dynamics is I actually have a negative trend. So I have negative correlation to their binding energies, which is not a great sign. The docking energies are so varied that while I do have a positive correlation, it's not great. In fact, if you look at the R values, they're basically random. So that's not much use. Right, what's next? I can still remember my chemistry lecturer explaining this one to me, and yeah, all of those assumptions were just garbage. So we needed to find a new way of tackling this problem. Docking scores. Docking scores are used to weight the conformations that the molecules are adapting and see which ones are favoured. It's a really good way of showing what the most probable conformation is if you know the binding site. But they're not very good for comparing between molecules. Usually they're additive. So if a molecule has more atoms, it will get a higher score. But there are ways you can game that system. Ensemble docking is one of those ways, and that's where, I think it's the next slide. Okay, it's the next slide. But the general gist is that you dock to a whole lot of different protein conformations. You dock to the same site, and what you're looking for is how does the binding score change as you change the protein conformation? What you'd really be hoping for is that a ligand has the ability to adopt different conformations to adapt to the change in the binding site. So one of the problems that we always have is we get presented with these beautiful crystal structures, which is this lovely static model, but the reality is they're under Brownian motion. They're all vibrating, they're all moving a little bit. So that conformation is adapting. So if you have a molecule that is incredibly rigid, then it needs to have a massive energy bonus to really stabilize that conformation. There are times where maybe you want a flexible ligand, so it can adapt to a whole range of protein conformations. But what you're really looking for there is that you get a nice constant score, so that even though the ligand is changing conformation, that the docking score stays consistent. You don't want to see a wide range in docking scores for different protein conformations. So here are 3 example protein conformations. So these are taken from a molecular dynamics simulation, which is where you're allowing Newtonian forces to act on the protein, and it's vibrating away. You raise it to a normal temperature, you give it natural motion. And what you do is you take a series of snapshots through that trajectory and you get different protein conformations, then you dock back into them. So what I've highlighted here is 3 areas where that protein, which is close to the ligand is in the thicker sticks, and what you're seeing there is the amino acid residues in the finer sticks. In green, you have the cartoon representation, which is sort of the secondary structure of the protein. And what I'm highlighting here is the changes that we are seeing in that secondary structure, which is derived from the changes in the conformation of those residues, and how that has changed through those snapshots. So what I ended up using was 10 snapshots from different simulations, which were the lowest binding energy of the ligand in that simulation. And I used it for all of the pharmaceutical compounds, all of the benzodiazepine simulations that I'd run. So I ended up docking into 40 different protein conformations, which I'm going to show you here. So what we managed to do was, from a panel of 200 odd terpenes that are known to be expressed in cannabis and found in cannabis, we found some compounds that were better in silico than linalool. This project is still undergoing work by our partner. They have completed the in vitro testing, and I believe the in vivo testing was scheduled to start at the end of last year. So it is quite possible that this product will be on the market soon with, unfortunately, again, can't say which terpenes. I'm very sorry about that. I'd like to thank the team. This is our founders and the rest of the team who helped throughout the rest of this project. I'd like to thank you all very much for your attention, and have a great day. Any questions? To GabaA, to where? Oh, it's going to be in a vape, so it's going to be inhalation, so it'll go straight into the bloodstream. I have no concerns about the bioavailability of them. I don't think any of them are going to be drugs of their own right. I don't think they're that potent. I don't think we're looking at benzodiazepine replacements, for instance. But I do think there is some value in giving GabaA a little bit of a nudge while you're also delivering CBD. I think that happens naturally with some of the other terpenes that are naturally expressed. But what I would say is that some of those terpenes identified in the 35 are very, very minor terpenes in cannabis. And what I expect is that there will be an artificially high level of these terpenes delivered in this CBD product. So we're enhancing what you could call, I suppose, the entourage effect. I'm not a huge fan of that. I think it is I don't think you need to put the 800, 900 compounds found in cannabis all into one particular formulation, but I think there are definitely minor components that you could amp up the concentration of and make a really wonderful product from. Thank you.